Today we are going to see the CBSE syllabus Unit 9, Chapter 11 Biotechnology, Principles and Process, and SCERT syllabus Unit 8, Chapter 4, Principles and Process of Biotechnology. Now the first question is, which one of the following is used for the production of SCP? What is SCP? Single cell protein. The options are given as Agrobacterium tumefaciens, Arthrobacter luteus, Candida utilis, Bacillus thuringiensis. See all the options. You have to find out the correct options from here. The correct answer is Candida utilis. SCP means single cell protein. Second question, how many base pairs are there in PBr 322 plasmid? This plasmid is nothing but which is act as a carrier for the foreign DNA or alien DNA. The correct option is, here 4 options are given. The correct option is 4,361 base pairs are there in PBr 322 plasmid. Next question is, TI plasmid is found in. TI means tumor inducing plasmid is found in. Options are Agrobacterium tumefaciens, second option E. coli, then third option Bacillus megaterium, fourth option Algaligenes eutrophus. The correct option is for the third one Agrobacterium tumefaciens. Agrobacterium tumefaciens. Find out the mismatch pair. You have to find out the incorrect matching. Dextran sulfate induce DNA uptake into the plant protoplast. Yes, correct answer only dextran sulfate. And also the polyethylene glycan also will be given here along with this. So these are used for the induction of DNA uptake into the plant protoplast. Second option, the bands of DNA in agarose gel is stained with eosin is given. We will see this option finally. Third option, northern blotting, that is transfer of RNA to nitrocellular membrane is correct only. Here we are transferring the RNA to nitrocellular membrane. Western blotting, transfer of proteins to nitrocellular membrane. This is also correct only. Then what is the incorrect match means? The bands of DNA in agarose gel is stained with not eosin, it is stained with ethidium bromide. Here the ethidium bromide should have come. So this is the incorrect match, that means mismatch pair. Next question, which one of the following is useful for the genome editing? Genome means collection of all the genes in an organism. Editing means some corrections can be done. What is the thing which is helpful for the genome editing? Here the options are BASTA is given. Next option is CRISPR-Cas9. Next option is DMH11. Next option is polyhydroxybutyrate is given. See all these things. The DMH11 is transgenic mustard. What is this? Transgenic mustard. The next one, polyhydroxybutyrate, is degradable biopolymer. This is degradable biopolymer. BASTA is nothing but the herbicide. But the question is genome editing. So the correct answer is CRISPR-Cas9. Correct answer is CRISPR-Cas9. Next question. Use of fungi to bring about remediation of environmental pollutants is use of fungi. See, the term fungi is very important. We will see all the options first. Bioleaching is given. Bioleaching means recovery of metal pollutants by the use of microorganism. Next one is phytoremediation means use of plants for the remediation of environmental pollution. Here we are using plants. Then bioventing means aeration is done to accelerate the degradation of environmental pollution. The next one is mycoremediation is the correct answer because here the fungi is used to bring about the remediation for the environmental pollutants. The first restriction endonuclease hint to always cut DNA molecule at a particular point by recognizing a specific sequence of how many base pairs. This is the endonuclease enzyme which cut the DNA. Restriction endonuclease enzyme which cut the DNA. How it will cut the DNA when it possesses how many base pairs? That is the question. So the answers are given here as 9 base pairs, 2 base pairs, 6 base pairs, 3 base pairs. The correct answer is 6 base pairs. 6 base pairs. Next question is, which one of the following chemical used to see bright orange colored bands of DNA when exposed to UV light? So bromine is given, sodium bicarbonate, calcium hydroxide, ethidium bromide. So here is this is the correct answer, ethidium bromide. Then only that can be seen under the exposure of UV light to see the DNA. Next question. Since DNA is hydrophilic molecule, it cannot pass through the cell membranes. So, in order to take up the recombinant DNA, that means plasmid DNA, the bacteria, that means a competent host, should be treated with a specific concentration of a divalent cation. What is this cation? Which increases the efficiency with which the DNA enters the bacterium. In the recombinant DNA technology, this is the plasmid DNA from which from the bacterium it will be removed, and the unwanted DNA parts, that means certain sequence will be removed from here with the help of the restriction endonuclease enzyme. And this is the foreign DNA or alien DNA that has to be cut, and it has to be united with the carrier DNA or plasmid DNA. So these DNA are called the foreign DNA, and this is the plasmid DNA. Plasmid DNA. So this particular part is removed. So this is the carrier DNA. With this we are going to joining this alien DNA or foreign DNA. For joining, what is the enzyme is needed? Ligase enzyme is needed. So with the help of the ligase enzyme, both are combined together. Now it will become the recombinant DNA. Recombinant DNA. This recombinant, as such, it cannot produce its colony or it will not replicate. So it has to be introduced into a host cell. So this is the host cell, maybe bacteria. When it has to be introduced into the host cell, DNA is hydrophilic in nature, so it cannot enter into this. So this host cell should be treated with the calcium. So this is the answer for this question. The correct answer is calcium only. Then only this DNA will be, recombinant DNA will be accepted by bacterium. Next question. For the repeated amplification of DNA, use of thermostable DNA polymerase is essential. From which bacterium it is to be separated? So, from which bacterium, the question is from which bacterium it is to be separated, this DNA polymerase enzyme. The four options are Thermus aquaticus, Escherichia coli, Lactobacillus plantarum, Clostridium botulinum. So the correct option will be Thermus aquaticus. This is the bacterial species from which the thermostable means it will be able to tolerate the temperature. So thermostable DNA polymerase enzyme is separated. Next question is, the ampicillin resistant gene is recombinant in recombinant DNA is called. Here four options are given here: origin of replication, cloning vector, recognition sites, selectable marker. Four options are given here. First we will see what is meant by origin of replication. This is the sequence from where the replication starts. Which one? The origin of replication is the small sequence from where the replication starts. This is cloning vector, you know well. I told you earlier that this is the cloning vector which carries the foreign DNA along with it. So this is cloning vector. The next one is recognition sites. Recognition sites, nothing but to the foreign or alien DNA, the vector needs to have very few, most preferably single recognition sites. Very few or single recognition sites will be there in the vector. Next one is selectable marker. This is the correct answer for this question. The ampicillin resistance gene in recombinant DNA is called selectable marker. So we will continue the videos for the next chapter in the next video class. Thank you, students.